So welcome to this October 2022 WAS mini conference, where we'll be mostly talking about queen issues and brood factories. And our first speaker tonight is going to be Dr. David Tarpy. Did I get the name correct there, Dave? And do you mind being called Dave or David, or what do you?
Yep, David is fine.
Okay. So I just want to do a little bit of an introduction because I think people deserve to know your very extensive background and all the work you've been doing for us over the years. So I'm going to basically just read a little bit here. And I want to say that since about 2003, you have been a professor and apiculture extension specialist at North Carolina State University. And your research has been ranging from very basic honeybee science and biology and all the way up to highly applied apiculture and bee management science. And your extension program has focused on information about honeybees and bee management for the public, for beekeeper groups, for local, state, national beekeeping associations, and you also administer a rather novel extension. Some of the two that I'd like to mention. One is your North Carolina State Queen and Disease Clinic, and also your online beekeeping education and engagement system, abbreviated BEES. And you have a series of online short courses, including an advanced queen biology and management. And I'm going to put up a link for people who haven't seen that. I'm going to put a link into the comments and invite them to check that out, because you have beginning courses, short courses, and then you have some more advanced ones, and that includes queen biology and management. Your research, David's research publications, have been cited 9,600 times by other bee scientists. And I was really, one paper I remember reading a long time ago, maybe when you were first getting started, David, was genetic diversity within honeybee colonies and how it prevents severe infections and promotes colony growth. And that was published in the Proceedings of the Royal Society of London. So that's quite an astonishing range and quite a commitment and quite a contribution to what we need to know about bees and beekeeping. So that leads us to your presentation today. If you're ready to go, you're going to be talking about diagnosing queen problems. And is it the queen? Is it the colony? Is it both? Or is it neither? So thank you very much for being here, David.
Yeah, thanks so much, Ron, for that nice introduction, and thanks everybody for being here today, and thanks also for embracing the kind of the new way of doing things and getting together on Zoom. You know, this has been really great to be able to talk to groups that otherwise travel logistics would make it impossible to be able to get together. Of course, you always miss all the sidebar conversations and all of the things that we enjoy in the real life meetings, but it really is great to be able to ease some of those bottlenecks and be able to talk to folks, in this case out in the Western Apiculture Society. So thanks for doing this kind of mini online symposium. I think it's great. I'm glad it's here to stay. But I also am glad that we are getting back together in other venues and that we're not just stuck in Zoom anymore. I think we're all welcome for that as well. So thanks for that. And you know, Mike Palmer, I have so much respect for Mike Palmer, and Mike and I have been kind of co-headliners on any number of different symposia and state meetings and other things like this, because, you know, I think we really complement each other when we're talking about queens and their biology and the practical management and everything. And so I think having these kind of different perspectives of a major issue when it comes to bees and beekeeping. And one thing that I've really learned and appreciated from Mike is really how nuanced all of this is. And as much as we know, there's still an infinite amount of what we don't know. And one of the things that, my goal here tonight is to, really talk about these supposed queen problems, or at least, what a lot of people are talking about as queen problems, and who is to blame. Is it the queen, the colony, both, or neither? And really, unfortunately, I have way more questions than I have answers with this. And so I really struggle with this, even with all of our research and talking with all beekeepers who are doing all this kind of boots on the ground, practical aspects of this. And it's not like I have answers. So if you're expecting answers here, unfortunately, you're going to be extremely disappointed. But I think in reframing the question and looking at it in perhaps a different light, it might enlighten us to really try to get to a better understanding of these queen problems. And so that's really my goal tonight. That would be, I'm really looking forward to the discussion afterwards of what people think of this. So when we look at, a lot of the Bee Informed Partnership surveys that have been done over the years, one of the consistently top issues that beekeepers are ascribing their colony failures, especially during the winter, are problems with queens, poor queens, queen failure. Only kind of weather and starvation and other problems rank that. But this is one of the primary management things that I think beekeepers have sovereignty over, trying to do something about these queens. And having a queen issue can then precipitate other problems that can then result in queen failure. And so there was a study done, Van Engelsdorp et al., that really showed that colonies that had queen issues at some point during the year were 3 times more likely to die during that year than those that did not have those problems. So it really is a significant issue if and when beekeepers have them. This is a separate study than that, also with Van Engelsdorp and Jeff Pettis, where we're looking at the importance of queens and especially their multiple mating that they have. And Ron, you were talking about, some of the studies that we had done in the past, looking at the importance of multiple mating, conferring genetic diversity among the workers. And this really kind of showcases that, where you look at the mating number of a queen. So some mating very few times, others mating with dozens of drones. And queens that mated with fewer than 7 drones were 3 times more likely to die than those within that first year, following them over the course of the season, than those that had mated more than that. And so having a well-mated, high-quality queen definitely has measurable and profound ramifications on the colony, even in the noisiest, commercial environment of these migratory commercial beekeepers. And so, these are real effects that are really invisible to us. It's really difficult to look at a queen and assess whether or not she is of high quality or low quality. And we'll talk a little bit about that. There's been other studies, a Trainer et al. Study that was looking at how the nest environment, and in particular the cumulative pesticides that are in the wax, that if colonies that had queen problems or queen events, requeening, premature supersedure, these kind of things, they had significantly higher levels of the cumulative levels of these compounds in the wax compared to colonies that were normal and queen right. And so, there's lots of different elements here when it comes to these kind of poor queens and what is prompting that. And so I just want to step back a little bit and talk about all of these different effects, things that can affect the queens. And I've just put them into, just conveniently, into 4 buckets, just 4 groups. And this is by no means comprehensive at all, but just looking at different biological aspects, chemical aspects, our beekeeping management, and then environmental aspects. So, certain biological things that can result in why good queens go bad are things like if she's diseased or not, her sperm viability of her stored sperm, all those kind of things. Chemical messaging and chemicals within the colony, such as the pheromone signaling or the pesticides and the wax, as I've mentioned before. Our management practices have an effect on queens, the acaricides that we place into the colonies, how we handle the queens, the different genetic stocks that we use. And then finally, outside environmental factors, such as nutrition and landscape intensification and all of those kind of things. And so there's lots of different things that makes it really, really difficult to pinpoint any one of them, because all of these other things are moving in real time and exceedingly difficult to control experimentally, right? But nonetheless, we're seeing a lot of these kind of bad queens or these queen problems. And if we go back to these surveys, these queen failures, I think that this is a very, very blunt and crude tool to be able to do these surveys, because there's many different things that are being ascribed to queens going bad. And so just for the sake of this presentation, I'm going to go over 4 different ones that I think are biologically and distinct, that may or may not have different causes or combinations of causes that result in them. So when we think of good queens going bad, we need to really think about, well, how are they going bad, right? And so for the purposes of this presentation here, we'll talk about the poor queens themselves, that is looking at the individual queens and their reproductive quality and or size, right? So kind of big, large queens versus small, runty queens. But that is distinct, I think, from queens going bad because they have a poor brood pattern, right? So that's kind of a separate diagnosis or way to ascribe that a colony has a bad queen. Not only the bad brood pattern, but what she is laying, specifically drone layers, right? I think that poor brood patterns can be different in many cases from the drone laying queens. And then finally, a lot of colonies are prematurely superseding their queens. So, queens are supposed to live 3, 4, 5 years. And I think anecdotally, at least, there's a lot of evidence out there to suggest that queens are not living nearly that long because they're being superseded when otherwise they all seem to be doing just fine. So I think kind of unpacking each of these things and discussing them, I think will be very helpful to try to give some nuance to this kind of poor queen definition that's just kind of thrown out there. So by looking at the queens themselves, just the physical entity of the queen, not the colony, but the queen, we know that there is variation in queen size and in their reproductive capacity. Normally within a colony, we see, female eggs or fertilized eggs developing into females of one of 2 types, either queens if they're fed royal jelly or workers if they're not. And so we see this kind of polar ends of a spectrum. But as those of you who rear your own queens and have grafted older worker larvae into those queen cells, you know that by doing that, you can get these intercasts, which are in essence worker-like queens or queen-like workers, because if you graft an older larva, then the reproductive quality of that queen is going to be a little bit less because she's already spent 2 or 3 days as developing as a worker, not being fed royal jelly, and therefore the queen is going to be smaller. She has fewer ovarials. She doesn't mate as many times. She's smaller. All of these things have been shown very repeatedly. So really, females are of this continuum. And within a colony, we usually see the ends of that spectrum, but you can manipulate colonies and you can actually make these intercasts or really, really crummy queens on purpose. And sometimes colonies will actually raise some low quality queens on purpose because they're in such a rush to make a queen very quickly that they don't care how good that queen is. They just want one as soon as possible. And so, queens from walkaway splits or other kind of emergency queen reared queens can sometimes, not always, but sometimes be of lower quality because of this kind of continuum of reproductive quality in queens. So, we did a study one time. This is when Juliana Rangel, before her move to Texas A&M, when she was here as a postdoc, we established colonies in 2-pound packages early in the spring on foundation strips. So they had to raise all, make all of their comb and raise all the bees and really stress them out to grow. But half of them, we had them headed by high quality queens, the best queens that we could make. And half of them were headed by low quality queens, those grafted from 3-day-old larvae. And then we tracked them over the course of, about 120 days. And what we found was that, sure enough, colonies headed by high quality queens had more worker comb, had a higher population by the end of the summer. They just tended to be better. But subtly so. It's not kind of night and day, but higher quality queens have a higher capacity to lay eggs, and so they tend to kind of accelerate in their growth a little bit more than those queens that are constrained because they're more worker-like. So again, this can be very invisible, and unless you do this careful side-by-side comparison of colonies, you might not be able to see it. But in the end, a colony is kind of an expanded trait of the queen herself, in that, if you have a low quality queen, the ceiling of your colony is only going to be so high. So again, it underscores the necessity of having a good high quality queen, one that's larger, mates with more drones, so on and so forth. But most of the time, I would argue, most beekeepers will go into a colony and they will say, this colony has a bad queen, not because of looking at the queen or carefully measuring her productivity over time or anything like that. They do so by looking at the brood pattern. And so when they see shot brood, we're all taught in beginner bee school that if you see shot brood, where on average every other hole, every other cell of what should be a contiguous area of capped brood, every other cell is missing, well, that's a queen problem. She's been mismated. You get rid of the queen, you get rid of the problem. That's what is the narrative that is often out there. So, there is some truth to this basis that, the shot brood can very well be caused by poor mismating. And this is how that is. It's because of a genetic trait that the bees have in how they determine a male versus a female. And really what it comes down to is that there's a single gene called the sex locus because it determines sex. And there are lots of different forms of this gene, up to, a couple dozen of these forms. And here they're just numbered 1, 2, 3, so on. And so when a queen mates with a male that has a different allele, a different form of this gene, when she fertilizes those eggs also have 2 different forms of that gene. 2 different forms of this gene result in femaleness, and therefore every egg that she lays is viable, and you get this really nice brood pattern, wall-to-wall capped brood. But sometimes, and this is where the queens don't know what drone she's mating with, sometimes she happens to be unlucky and she mates with a drone that happens to share one of her 2 sex alleles. So that in half of the eggs that she fertilizes with this drone, they are homozygous, meaning that they have 2 copies of the same type of gene. That confers maleness. You've all heard the term before, diploid drones, and that's what this is. It's a fertilized egg, but it still develops into a drone. That drone is kind of a genetic anomaly. It will be infertile. They're not going to be able to mate. And so right as that egg hatches, the nurse bees come along and they cannibalize those larvae. The queen's going along and she's fertilizing these eggs, thinking she's, laying all of these workers, but half of them are killed. And so this is what leads to shot brood. This is what leads to this really poor brood viability within the colony, because she has this genetic mismatch of who she mated with. And because queens don't remate throughout their lifetimes, then it's true. The best remedy for this type of situation is to get rid of that queen and put a new one in there. So there's this linkage then between a poor brood pattern and a poor queen. So we see a poor brood pattern, we get rid of the queen, move on. The problem is that when you go into a colony, you can see a brood pattern for different reasons. So here's a bad brood pattern, but is that because of the queen? For those of you who are studying for your master beekeeper exams or whatever, you will know that this is telltale signs of American foul brood. Well, that has nothing to do with the queen's mating, presumably. And, you see these perforated cappings and, hint of a scale. Yeah, that's going to be a different reason for a bad brood pattern, similar to this. This is European foul brood, bad brood pattern, but it's not the queen. Chalk brood, bad brood pattern, same thing. And even our favorite, parasitic mite syndrome or idiopathic brood disease syndrome, which is in essence parasitic mite syndrome without the mites. You see this manifest in the colonies as bad brood patterns, but it's not because of this genetic trait of queen mating. And in fact, it has to do with pathogens and parasites. It has nothing really to do with the quality of the queen. Yet we still kind of go into these colonies, and if we're only doing a very kind of cursory inspection, we see a bad brood pattern, we blame the queen, and no further inspection is needed. And so if there's anything that I want you to take away from this tonight, is that if you see a bad brood pattern, you have to rule out all of these other things before you blame the queen, because you need to make sure that you don't have, huge levels of mites and the pathogens that they're vectoring or any of these brood pathogens, because it's really easy to blame the queen and not do a deep dive or investigation. It's really imperative to make sure that you go through and you ascribe why that colony has a bad brood pattern. Now, several years ago, we teamed up with Katie Lee, who was then Marla Spivak's PhD student at the time. And, we were, again, interested in this, idea of what makes a bad queen bad and what makes a good queen good. And of course, with Katie working with a lot of the Midwestern commercial beekeepers, she's in a lot of hives and they're seeing a lot of colonies. And so their beekeepers would often find these poor queens as defined by those colonies having a poor brood pattern. But what was really great about Katie is that she was able to convince those beekeepers to go to the hive next door with a colony with a good brood pattern and pull that queen out too. And so she would send us these pairs of queens, and we would measure them for their body size, their sperm count, their sperm viability. We measured them for all sorts of different viruses and pathogens. We took samples of the workers and we measured the pathogen loads of the colonies. We measured, we took wax samples and we measured like 200 different pesticide residues in those colonies. And we got dozens of these pairs of colonies from lots of different operations. But unfortunately, we didn't see a single smoking gun. There wasn't something in the bad queens that was not, in the good queens or vice versa. In one case, in one operation, it seemed like the poor queens had a high level of deformed wing virus. In another one, there seemed to be a higher level of a certain pesticide compared to the good ones. In another one, it seemed to be a sperm viability issue, but there was nothing consistent. So we were kind of disheveled about that. And for the second year, we were going to do this and we're going to kind of double the sample size and maybe hopefully find something. When Katie, I think in a real brilliant move, said, well, rather than just sampling the queens, why don't we switch them? Why don't we take the queen in the colony that has a poor brood pattern and put it in the colony that has a good brood pattern and vice versa. And so if it's the reason, if it's the queen that's resulting in that bad brood pattern, then this colony, after a single brood cycle, 21 days, their brood pattern should suck and go down, whereas the colony with the poor brood pattern, their brood pattern should improve, right? And then we sampled them and measured these variables again. And in essence, what we found is that it was not really the case, right? That there was very incremental, non-significant decrease in the good brood pattern or increase in the bad brood pattern. So it really wasn't the queens per se. So again, this underscores the idea that we're assessing the queen based on the brood pattern of the colony. But if you think about it, that's a really bad way to assess the queen herself, because does the queen raise those brood? Of course not. It's the nurse bees that do. So it's a colony level trait, not a queen level trait. And so we really need to disassociate this whole thing about a bad brood pattern being the queen. And instead, we need to do a lot more sleuthing and doing a deep dive into trying to ascribe the reasons for those bad brood patterns. Because yes, it can be the fault of the queen, but more often than not, it is not. And so we need to kind of disassociate those things. Now, as I said before, there's these 4 different things that we're looking at. So it's not necessarily just the shot brood pattern, but the fact that the queens are sometimes laying drones when they otherwise should not be, which is an indication of poor insemination. The problem with seeing lots of drones in the brood nest is that, again, it's nuanced. There are different ways that you can arrive at that. One is a queen problem like this. For those of you who kind of recognize this, you see multiple eggs per cell. Most of them are laid around the side. This is telltale signs of a queen problem, but not what you think. This is a queen problem in the sense that these are laying workers, and so they haven't had a queen for like 2 or 3 weeks. And the colony is kind of on its last gasp. This is their kind of dying breath, right, of the workers developing their ovaries and laying unfertilized eggs that become drones, and they will rear them. And so you can see lots of drones in the brood nest and just chaos ensues, but it's not because of the queen per se, it's because she hasn't been around for a long time. So you need to make sure that you can distinguish lots of drones in the brood nest because of laying workers or something like this. When you see something like this, what you see is some worker brood, but then these domed drone cells where the worker brood should be. And when you see that just a little bit at a time and then with increasing frequency, this is often an indication of an older queen where the sperm in her spermatheca are dead, or she's running out of live sperm. So again, the queen's going along, trying or thinking that she's fertilizing these eggs, but she's starting to shoot blanks, so that she has sperm in the spermatheca, but it's either all dead or she's already used up most of the live sperm. And so she can't fertilize those eggs. So that's how you can tell, that the queen is running out of gas. She's running out of sperm. Versus a third time when you can see lots of drones in the brood nest. And that's something like this, where every single egg is unfertilized. Every single brood in the center of the worker brood nest are actually drones. This is more indicative of the queen never having sperm, usually in young queens, newly mated queens, that actually failed to mate, right? So you have to be able to distinguish among these things. You have to know the age of the queen in the colony. You have to be able to read the frame, and you have to, be able to suss out the brood pattern itself in order to see what kind of drone layer you have, because not all drone layers are the same. Now, I just threw this in here because I find it really interesting, although it's kind of more anecdotal than actionable. We were doing an experiment where we actually wanted drone layers, right? We do all sorts of crazy stuff as scientists. And so we were soliciting drone layers from commercial beekeepers, and we wanted them to send pictures of their drone laying queens and their brood patterns before they sent them to us, and just to verify that they were. And so they, put them in cages and shipped them to us in battery boxes or whatever. And, some of them we dissected them right away, and others we threw into colonies so that they would lay drones in worker-sized cells. That was kind of why we wanted them. But there were a couple very interesting things that we found. One is that about 40% of the queens that we got were indeed drone layers. They were drone layers when the beekeepers saw them. There were drone layers when we hived them up. They were drone layers. Interestingly, 20% of them, they started laying drones, but then after about a week, they started laying perfectly fine fertilized worker brood. And then another 40% of them, we hived them up, and they were beautiful laying, workers from day one, even though they were drone layers in the colonies where they came from. So this suggested to us that, drone laying, it's supposed to be, oh, they run out of sperm or all the sperm are dead, those queens are no longer good. Drone laying may actually be a recoverable condition, or it might be an environmental condition. Something, for example, like a blockage of the spermathecal duct, so that, the queen gets a lot of nosema, she's trying to release the sperm, but can't, and so it turns out to be an unfertilized egg, but she's cured of that infection. Now all of a sudden she can start fertilizing eggs again. So again, it might be that drone laying is recoverable, but we need to do some more investigation into that to see if it's really worthwhile trying to rescue them or not. Anyway, I just thought that was an interesting side note. All right, the last issue that a lot of beekeepers are seeing are premature supersedure. And, this, I'm kind of throwing swarming in on this too, where, colonies can have problems by having an unexpected kind of turnover from mother queen to daughter queen. A lot of times this happens with swarming, which I'll go into, but also supersedure, which I'll end with. But when colonies swarm in the spring, if you're not on top of it, every 7 to 10 days and going into the colony, it can really cause problems for the rest of the summer. So, if you're not on top of it in swarming season, you can have real hard time trying to understand what's going on. And you got to remember the timeline of, kind of the queen development, so that, if you don't go in every single week, you might have capped queen cells where the colonies swarm on you and the mother queen's gone and no longer laying eggs. And so what happens is that you start to go through this period where there's no eggs or young larvae, and then eventually all the capped brood hatches out as well. And so if it's 4 weeks, since you last checked your colony, and you go in and all of a sudden there's no brood at all, well, you think, oh my gosh, I need to throw a new queen in there because, they've gotten rid of their queen. But actually what's going on is they were raising their own queens, and there's a virgin running around in there, and you don't realize it. And so now you're throwing your money down the drain because you're putting in a mated queen, and there's a virgin queen in there, and she'll kill her. And, you just wasted a perfectly good queen. So you really need to remember how long it's been since you've been in your colony, and think about the timeline of when virgin queens will come online, when they will start mating, and when they'll start laying eggs, and then when you'll start seeing brood again. But if it's been 6-7 weeks since you've been in your colony last, and you still don't see any brood, or at least eggs and young larvae, then boy, you better do something quick, because you might develop laying workers very quickly. So again, if you're not on top of things during swarming season, that can lead to these cascading problems where the colonies are going to kind of go into these brood dearths, and the strength is just going to really plummet when they should be going up. And so it's just imperative that you stay on top of the swarming and going into the colonies and being able to read the different symptoms, read the frames. If you see something like this, here's a great example of a capped queen cell. Well, right away that there's this little perforation in there, so that this queen is about to emerge, right? So you kind of know the timing of it. If you see a whole bunch of them and none of them emerged, then that you're still pretty early in this, but the mother queen is gone, right? If you see something like this is a great way to read the frame, where here you have an emerged cell because she chewed her capping out. It's this perfect circular capping at the entrance. She emerged, and then she seeked out all of her rival sisters, and then she chewed a hole in the side and then stung the occupant before she had a chance to emerge. And you can see that because they have their cappings, but the workers came along and tore down the rest of the cell and got rid of those dead rival queens. So that there's at least one virgin queen on the loose in a colony like this, and at that point you might as well just shut it up and let her go mate and take over. There's no point in trying to requeen this colony at this point. Again, an ounce of prevention is worth a pound of cure. When you see something like this, when you see all of this there's no brood in the colony, but the workers have really cleaned out and polished the center of that brood nest, that's often an indication that this virgin queen has mated and she's just about to come online. These workers are excited. They've prepped the nursery as best as they can. And pretty soon you're just going to see all of these cells just laden with eggs. So you're only days away when you start to see something like that. So again, you need to read the frames and realize kind of what stage you're in this kind of intergenerational time. But again, that's something that you can prevent. Prevent swarming can help with all of these types of problems. What I think is a lot more difficult is how to prevent supersedure. And this is something that I've struggled with because I just don't understand how workers are able to decide that a queen is bad and they want to get rid of her. To try to compensate for this, I've come up with a framework to be able to try to think about it a little better. Now, a lot of people will say, well, if you have a poor queen, small, runty, hasn't mated with a lot of drones, those are the ones that are going to get superseded and the good queens are going to stick around. Well, I'll go back to this study with Juliana, where we looked at these colonies headed by high and low quality queens. And in fact, the low quality queens were not replaced more frequently. In fact, 2 colonies headed by high quality queens were superseded, 2 colonies headed by low quality queens were superseded, and there wasn't a difference between them at all. So the workers seem to be queuing in on something else that is not kind of what we think is a good versus bad queen. It's something else. And so, again, I came up with this framework of how these workers are able to collectively decide, okay, we're going to get rid of this queen. So bear with me here. I'll just kind of walk through it. But I do so by breaking down the life cycle of a queen into 4 groups over time, over her lifetime. So the first phase of a queen's life is the development phase. So this is where they're being reared, and then when they hatch, and then they mate, and then they start laying eggs. So that's the kind of only a matter of a couple days or weeks during this development phase. Once she starts laying eggs, the queen then enters what I call the establishment phase, which is where she's introduced into a colony, or she starts laying within a colony. And after about 9 weeks or so, the entire worker population has turned over, where the worker offspring from the previous queen, whoever it was, mother, foreign queen, whatever, they're all died off, and then the colony is now all of her daughter workers. So that establishment phase goes from introduction to the complete turnover of the workers. The third phase, oh, sorry, and then during the interface between these two phases, the development and the establishment phase, is where we, transport, ship them in packages, put them in battery boxes, introduce them to foreign colonies, all of that. So there's a lot of management that goes on during this interface between these two phases. The third phase is the survival phase. And so, after the population turns over, we consider the queen fully accepted, and we want to maximize this survival phase, right? 3, 4, 5 years. So this is what beekeepers want to get the most out of their queen, right? But even the best queen, the longest lived queen, they're not immortal, right? So eventually they're going to senesce. Eventually, they're going to go downhill, hopefully after a matter of a few years, and enter the fourth and final replacement phase, where the queen starts to fail by any number of different mechanisms, and eventually the colony will replace her through supersedure. Now, one problem that's out there is that colonies supersede their queens, but their requeening success is not perfect. So sometimes we see this in different operations where the colonies go to supersede their queens, but then they tear down all their queen cells, and now the colonies have no options. And I don't know what's going on there, but again, it's not always perfect. But if it does work, then the cycle just repeats itself. Then they get a new rearing of a new queen and the next generation of queens. Okay. So if you follow me so far, hopefully you have, you will see, we can see that if we just look at the sperm and the spermatheca. So obviously as queens are being reared, they're still virgins. They have an empty spermatheca. Once they mate, they have a full spermatheca. And then over their egg-laying lifetime, they're using the sperm from their spermatheca, so that useful amount of live sperm in their spermatheca depletes over time. Now, not all sperm in the spermatheca are viable, so some of them, are not alive. So, really it's a complicated function of, the viability and the egg usage and how much sperm she started out with, and so on and so forth. But again, eventually, the queens reach a certain point where they run out of sperm or they become unacceptable to the workers. So there's some sort of perception by the workers where beforehand, okay, she's good, she's good, she's good. We perceive her as being good and viable. But at some point, their perception of her, their opinion of her, right, the opinion poll of the queen crosses some sort of threshold where she's now unacceptable. Because the workers have to determine, it's a binary choice. It's either you keep the queen or you throw the bum out, right? It's a binary thing. And so there has to be some sort of threshold where all of a sudden this perception tanks and now she's unacceptable. And that's when they engage in supersedure behavior, rearing new queens, starving the old mother queen, whatever it is. It's still very foggy as to what happens during natural supersedure. And what I would say is that's what's happened, or what seems to be occurring more frequently, or at least we're being able to see it more often, is that this threshold is crossed earlier in the queen's lifetimes during the survival phase, so that queens are being superseded after 1 year rather than 2 or 3 years, or even during the establishment phase where people are establishing packages with newly mated, seemingly perfectly fine queens, but they're being superseded by the workers because they're deemed unacceptable, and we don't know why. So I think there's some interest here in trying to kind of tease some of these different things apart. But again, it's very complicated because, again, there's all these different factors that go into the worker's perception of how good that queen is. And I'll go back to those different biological, chemical, management, and environmental factors that are all interacting with each other and very hard to control. and they could be affecting the queen, the worker's perception of the queen differently during these different phases of her, lifetime within a colony. So again, it makes it really, really difficult. And I don't have any answers here, just more questions. But I think by thinking of it in this way, we can start to better wrap our heads around it and realize that, not all queens are the same, not all colonies are the same, and that they might be treating them differently in these different phases throughout their life cycle. So to wrap up here, hopefully we'll leave enough time for some questions. There are all of these different things that can be reasons why good queens go bad, and we can either blame the queen, or we can blame the colony, or we can blame both. And I would submit to you that some of these things are in fact something that is the fault of the queen, but most of them are a combination, if not really largely a factor of the colony more than it is the queen. So we need to get away from this narrative, especially of looking at the brood pattern as a queen problem, but more of a colony problem, more of an environmental problem and a collective problem. And so if we look at it that way, then we might be able to help rescue some of these poor queens, because it's not the queen herself, but really the environment in which we happen to find her. So with that, hopefully we have enough time for questions, and I thank you for your attention.
Hey, Dave, I have a question. Can you
Yes.
hear me?
Yep.
Yeah, you mentioned a queen failing to get eggs fertilized, and mentioned nosema in the oviduct. I've never heard that mentioned before. Tell me what you know about nosema clogging the oviduct.
So I'm not saying clogging the oviduct. It could be in her, obviously it's in her mid and hind gut, but it might be descending the gut so much that it kind of kinks the spermathecal duct or something like that, right? So yeah, it's not an infection in the duct per se, but there's something going on, or it could be environmental, right? Like maybe these colonies where the queens were laying before, the wax was so toxic that, that it was like preventing fertilization or something. And when they're put in a different environment, then it's fine. So there's lots of different things.
Okay,
But
I understand.
Just the fact that you could remediate a drone layer is kind of crazy to me, or at least I don't know, maybe other people have seen this for a long time, but to me that was a real eye-opener that it's not a fatal condition perhaps.
Right. And over the years, that's what we found. We rarely reject a queen. We always give her a chance to recover. A lot of times a smaller colony, you just combine two small colonies and all of a sudden everything turns beautiful again. A queen that you thought was doing nothing turns beautiful. That's happens so much that we just rarely.
So that's par for the course in your operation then, Randy.
Absolutely.
So what do you think then it might be kind of causing that kind of temporary infertility?
I have no idea, but with our nukes, we see it. It's not unusual to see that, see them recover. Well, the big question I have is the failure to successfully supersede. Supersedure, if a queen's starting to fail, back in the day, we rarely had a colony go queenless. And we don't use synthetic miticides, and we're not in much agriculture. So I can rule out pretty much pesticides in our operation. But ever since Varroa arrived years ago, we have a much higher rate of what I call unsuccessful supersedure. That you used to, back in the day, 50 years ago, you could get a hive of bees put in the backyard and just never open it for 10 years. That's right. and you still have a colony of bees in there. It would be the same queen. They might replace the queen every year. That's not the case anymore. And I have no idea what it was a virus issue or whatever. It seemed to correlate with the arrival of Varroa. So that's why I'm suspecting a virus possibly. But yeah, that's what I pull my hair out on that one.
Well, so that's also why I'm really couching it in terms of the worker's perception of the queen.
Yeah, or
Because
failure of the
we're always
workers
thinking the
to realize.
signaler. We'll always think there's something wrong with the queen. But if something is wrong with the workers being able to smell the queen properly or, detect her, if that starts to get skewed up, it has nothing to do with the queen. It's if something happens to the workers, then all of a sudden they can make dumb decisions, right? Like supersede the queen and then tear down all the queen cells, right? That's a dumb decision, but it might be their perception and their regulation of that process that can be screwed up. Not so much the queen. It has nothing to do with the queen, right?
Yeah.
So
evolutionarily, you'd expect the queen to put out a true signal before she failed. Otherwise, she loses her line, her genetic line. Yep. So evolutionarily, it'd be the advantage of a queen to tell the workers to supersede me, and colonies will often leave the mother and daughter lay together for a while. It's not like it means the death of the queen. So I'm wondering whether the queens are no longer putting out a true signal to the workers.
That's possible, but also in all communication, you have the signaler and then you have the receiver.
Oh yeah.
So again, we're often thinking of it on the signaler side, but not as much thinking of it on the receiver side. So yeah, we have done some work looking at high and low quality queens, again with Juliana, and yes, the pheromone of low quality queens are not as good as the high quality queens. So there's definitely something there, but they still smell like queens, maybe not kind of as robust. So it's subtle. And so the question is that subtle difference enough to have a really profound difference among the worker receivers?
Yeah, but the question I have is that the workers get tricked, or not tricked, they don't recognize the queen is failing, not recognizing the queen's failing. And so you get to the point where the colony is unable to supersede the queen because the queen stopped laying fertile eggs at that point. The other thing I noticed is that when the colonies are stressed by whatever, when trichomite first came, with varroa, with thymol, anything that's stressful to a colony, the bees will often blame the queen for the stressor, for something going wrong, and they will supersede at that point.
So that's a great point, Randy, because think about it, right? Okay, we're about to go into a midterm election, right? But like during presidential elections, right? We don't like the government, so you throw them out, right? Or you like how things are going, so you keep them. We have this binary choice when it comes to the presidency, right? You either keep them or you throw them out. And that's all the workers have too. It's like, hey, we're stressed. This is a bad environment. We can't do anything about it, but we can get rid of her, right? And so like all of these different wires are being crossed. And I think that you're right. The more stressed they are, the more they take it out on the queen, and it has nothing really to do with her, but it's the environment. So that's. So I know there's a lot of other questions in the chat here. Do you want me to go through those, Ron? Or how do you want to, or you want to ask them and I can.
Yeah, why don't I go ahead and do that for you? So, and then you can concentrate on the answers here. Okay, I have a colony superseding the queen a couple of weeks ago. Temperatures already 42 Fahrenheit. Do bees take into consideration drone population and temperatures? I guess that's in reference to the supersedure. Or are they dumb and supersede regardless? This queen may not be properly mated at all. Thank you.
Yeah, so I think that gets to a lot of the discussion we were having with Randy there, that I don't think bees are dumb, but I don't think they always make great decisions, right? That's a big difference. And so, yeah, they might supersede because they think that's the right decision to do right now. But in the end, that queen, if they do make a supersedure queen, the drone populate, they are not trying to kind of gauge what the local drone population is. They are not kind of doing that kind of calculus. And so they might wind up with, a not very good queen at the end of all of it, right? So they're not dumb, but they are, that doesn't mean they always make great decisions.
Or it might mean that we don't really know what is smart and what is dumb from the bees' perspective.
Correct. Or at least we don't know what are the things that they're making this collective decision. That's my point. And that's what I would love to just focus on this for the rest of my career, because it's so complicated. And it's kind of mind-boggling to try to get to the end of that.
Oh, it's fascinating. The same person has asked another question here and wanted to ask your opinions and concerns about artificial insemination in general, as well as specifically the person choosing the drone sperm instead of natural mating by the faster drones able to mate with the queen. Okay.
Yeah, so there's no evidence that faster drones have more or better sperm. That isn't to say that isn't, but there isn't any evidence to that effect. And, I think it all comes down to the inseminator and how choosy they are of the drones. If they're very selective and raising good, high quality drones with good nutrition and, of the right age and all of that kind of stuff, I think that the instrumentally inseminated queens can be on par with naturally mated queens, but we're not all that good. And I think there are many more ways that things can go wrong. So yeah, naturally mated queens do tend to, in my experience, on average, kind of live longer and just have a hardier, more diverse set of drones that they're inseminated with.
But the question fundamentally has a really interesting perspective, and that is we're making these decisions, and we're kind of interfering with what might be the natural order. And until we know more, I guess we don't know always that we're making the right
No,
decisions.
that's very true. There's also there's an implicit assumption there too, is that nature is always best. And as we were saying before, that they're making the best out of a bad situation. It's not always perfect, right? Nature is not always perfect. It's just better than those who didn't do as well, right? So I think, there's very little control, at least as far as we understand, as far as queen, they don't make a choice of who they mate with, that it's kind of a stochastic, there's a randomness to it up in the cloud. So there's a randomness to it when you're choosing drones for II as well. So to me, I'm not too worried about that. But again, bee biology can always be surprised.
That's for sure. And even with us humans, sometimes us inferior humans get lucky. So anyhow, Jessica G, I hope that I turned on your ability to talk, because we need a little more information about your question. Is this a function to prevent repetitive inbreeding? And you probably made that comment during a certain part of Dr. Tarpy's talk. So
I bet I can guess which one.
Oh, okay.
And that was probably when we were talking about that single gene locus that results in inviable brood. And so your answer is exactly right. Yes, because if a queen mates with a brother, they have a 50% chance of having the same sex allele, which results in that shot brood, inviable diploid drone brood. So they actually mate with 1 to 2 dozen drones to ensure that they don't mate with all brothers, or at least that they have, mate with enough drones that have different sex alleles than they do, so that their brood viability overall is higher. Even if they mate with 1 or 2 brothers, that's kind of swamped out by mating with so many drones. So that's exactly right and very insightful of you.
Great. Okay, your next question, Dr. Tarpy. I had a queen this year that laid a perfect pattern of eggs for 3 weeks, but I never saw larvae hatching in the nuke. Lots of pollen, I was feeding syrup, I swapped her with another nuke, and she did the same thing in the new nuke. And the new queen laid successful brood in the original problem nuke. Any idea what could cause 100% rejection of her eggs?
Well, I said that I would entertain questions, not hard ones. So move
well,
on. Excellent.
There's kind of an addendum to that question. Do you see that
Yes.
there, right?
Yes,
sorry?
I think the addendum to that question is I should add the egg laying rejected eggs was one of the first rounds of new queens grafted from a new breeder queen. Okay, I'm not quite following that.
Instrumentally inseminated queen.
Yeah.
Oh, okay. Yeah.
No,
yeah,
you got me. I still wouldn't know why you would have 100%. I know that newly inseminated or newly mated queens can often what I call pop the clutch, right? Like they can get really over anxious. They need a little practice laying eggs. Sometimes they'll lay 2 eggs in a single cell, and they can do, like a teenage driver with a stick shift, right? It's just not quite clean and smooth, and then with a little bit of practice it smooths out. But why the eggs would be cannibalized wholesale, yeah, that's beyond me. I'm sure others might have some ideas, but I've never seen that, and I can't offhand think of what might be a reason for that.
So we're assuming that those eggs were not viable, never hatched, and then they were
Yeah.
pulled out. Yeah,
Yeah, so there might have been a wholesale, like not just the sex locus, but something that was totally fatal. But again, if she kind of overcame that eventually, then that's even more weird. So yeah, I'm not really sure.
Mhm. What about the 10% or so of colonies that have a mother-daughter queen combo?
Yeah.
Have they decided the mother queen is bad, and did an event occur that they thought they should replace her and then they changed their minds?
Again, more questions than answers with all of this. That yes, oftentimes if you see one queen and you keep looking, you can find a second queen. We don't even know in supersedure how the old mother queen dies, right? Does the daughter queen eventually kill her? Do the workers kill the old mother queen? Does she just get neglected to death? That's what I think is happening, is that she just senesces, and she just can't eat anymore, and she just dies of old age, right? But like nobody's ever been able to document that because we can't impose it, right? This is just something that happens really naturally but rarely, and so being right there at that time and place can be really tough to see it with regularity enough to be able to measure that. And so yeah, I just don't know the answer to that. They're doing it. It's nice when colonies have those overlap of generations, right? Because then the colony itself doesn't skip a beat. But why don't all colonies do that, right? Why do some of them have this gap or a hiatus? Why do they not have multiple daughter queens, right, as insurance policy? I just don't know.
Yeah, I guess there are a few things left for you guys to figure out. This is similar to an earlier question that was asked by the same person, and that is, do worker bees, I keep jumping here, sorry about that. Do worker bees consider drone population or temperatures before they start superseding? Or do they just kill her regardless of timing that could lead to the colony to their doom?
Yeah, I think we were talking about that before, is that the workers are not assessing the local drone population or anything like that. They're going by kind of the internal colony stimuli, whatever those are, and making the decision based on that. Whether they get below that threshold and they decide off with her head, we don't know what goes into that. But I really don't think, it might be temperature or something like that, but they are not assessing how many drones are out there. No, I can't see how they would be able to do that.
Well, and I'll just pipe in here briefly. It makes me kind of think a little bit too about laying workers. Like, do they know that their eggs are not going to be viable? Potentially not. They still want to lay. There's some sort of mechanics in there that sort of inspires that, even though from the outside we're like, that's, why would they spend the energy to do that?
Yeah, no, to me, that's like a evolutionary vestige, right? Is that the workers, all of the bees used to be reproductive. And then when they evolved sociality and then this kind of repression of reproduction, that's the evolved situation. So when you remove that, then they go back to their basal instincts, which is to reproduce. The problem is that they can't mate, therefore they can't fertilize the eggs. So really, I think that's more of a not a drive to rescue the colony or anything like that, but it's just that they've been alleviated from this repression and they're just resorting to their basal instincts.
Melanie, you're making the assumption that the eggs are not viable. They are viable.
Well,
They still raise drones.
Females,
If the colony is going to die, the only chance they have of getting their genes in the next generation is to raise a small drone that might have a chance of mating with other queens. So it makes total sense evolutionarily. It's a last gasp attempt to get your genes in the next generation.
well, and yeah, no, I'm glad you pointed that out, because that is true. I made that assumption that they're not viable, meaning that they're not necessarily producing fertilized eggs. And I don't want to get us off on a tangent, but it has me also thinking like, okay, what about the Cape bees? You know, what's that sort of evolutionary instinct and how they've made that work without having to mate. So
yeah, actually my mentor was from South Africa, a guy named Dave Fletcher, if anybody remembers him from the 70s and 80s. And he was there. He studied the Cape bee a long time ago, and his contention on that is that it's so windy on the Cape and they have a lot of queen loss of flying virgin queens that the Cape bee has been selected for thalidicy and workers being able to fertilize, quote unquote, fertilize eggs to make new queens because they're in this kind of very treacherous environment for mated queens. So that was his evolutionary explanation for that. Impossible to test, of course.
Makes some sense. Yeah, thank you. Can you speak a little bit about the idea that supersedure sometimes may be caused by the queen's pheromone being masked by some of the chemicals we put in the hive, particularly mite treatments?
Yeah, sure. So I also want to make sure this is the last question, because I want to hear Mike's talk and give him the floor as well, and not to dominate this. But we don't know much about how miticides or really any environmental chemicals or contaminants are affecting the perception and the odorant binding proteins and other things of the workers' antennae and their ability to smell. We don't really know much of that at all. Now, maybe toxicologists kind of can work through a lot of that in a generic way, but practically we don't have a lot of data on that. So it's an open question, and I think an empirical one and an important one. So it's very possible that we're kind of deafening the ability of workers to smell the queen properly, and they're taking it out on the queen. It's possible that it's totally benign and it's something else, but I think those studies are deserving of some careful study.
Fantastic.
So with that, thank you guys so much. It's a great conversation. I really appreciate liaising with all of you.
Well, we appreciate that you were able to do this. Thank you so much. Thank you.
Yeah, and Dave, if you have a moment and are up to it, that you maybe can look at a few of the other questions in the Q&A. But yeah, let's listen to our next speaker.
Okay, yeah, sure, we'll do.
Okay, and our next speaker is going to be Mike Palmer, and I just want to he deserves this sort of an introduction too. And so his background is that he grew up near New York City in the suburbs, and from childhood he had been fascinated by all sorts of plants and everything around him, insects, animals. He went up to Vermont to study, and while he was there, he met and fell in love with the local scenery, which included his wife, Leslie. So he stayed and started beekeeping about 1974, he says, with limited success or varying success. That's about 50 years ago. So he's got a lot of experience to share with us. By 1998, with the mites and other issues, he changed the way he overwintered his bees and concentrated on the quality of his bees and not the quantity of the bees. Today, he raises about 1,200 queens and manages over 1,000 colonies, and he is recognized as a worldwide authority on sustainable beekeeping. He's built up his French Hill apiaries into a very successful farm. He's known for his progressive beekeeping, and his a lot of people will have met him indirectly on YouTube. Some of his presentations have received over 100,000 views, which is absolutely phenomenal. So today, Mike is going to be presenting brood factories, which I suspect is going to be more interesting than a talk on honey factories. So Mike, if you're ready, you can get in there and share your screen if you want to, and you've got it. Okay, thank you so much.
So yes, I guess I'm not muted. So hey, so nice to be here. Gosh, there's really a lot of names that I recognized in the participant list, you know. And I got to mention David, you know, the first presentation I ever did was in 2006 in Massachusetts. I didn't have a clue what a PowerPoint was. And, you know, and I just winged it. And who was there as my co-speaker? Of course, David. And how many for years afterward, I'd show up at a meeting someplace and who was the co-speaker? David. You know, I remember one in, gosh, where was that? In Pennsylvania, was at the Quaker School. Remember the Quaker School, David? So, you know, it's pretty amazing. So it's real nice to see you here again. It was great to catch up with you at Ithaca. So anyway, I'll get right on to this. This is a presentation is about brood factories. You know, what is a brood factory? Well, a brood factory is a hive of bees from which you harvest brood, combs of brood to be used somewhere in the apiary for whatever the use, for whatever you need them for. And the brood factory, its whole purpose in life is to make frames of brood. You know, it's not a honey producer, you know, it's a brood factory. Can any style be of beehive be a brood factory? Yeah, of course. You know, you could use 10 frame lengths, you could use Ware hives, you could use top bar hives, you can use nucleus colony style hives, as I do. So it doesn't really matter the style of the beehive. Yes, they can all be used. For instance, Brother Columban at Buckfast Abbey in 1900, Brother Columban was Adam's mentor. In 1900, Columban devised a beehive that would hold 4 nucleus colonies. And in the springtime, he used the excess brood in those nucleus colonies to boost his honey production colonies to make a better honey crop. Brood factory, same thing in 1900. And I'm sure if you were to look back if you could go back in time, you'd find other beekeepers that did something similar. They devoted certain beehives to making brood for their apiary. Are brood factories and resource hives the same thing? Well, you know, I suppose they are. Are all resource hives brood factories? No. All brood factories are resource hives? True. Are all resource hives brood factories? No. Part of the problem is, and in the videos I've seen of people talking about how to use resource hives, it seems like the most common use of a resource hive is to have an established small colony within the apiary with a queen right colony, that if something happens to a colony in the apiary, you can rescue that colony with your resource hive. So it's a one-to-one thing. You have one resource hive, you save one beehive with that resource hive, and now the resource hive is gone. Well, I hope by the end of this presentation that you'll realize how much more brood factories are, how important they are in my apiary, and sure they can be in your apiary as well. And let me just say, this is something I kind of been working on. Originally, I had this triangle. Was the corners of the triangle were mating nuclei, nucleus colonies, and production colonies, and I showed how each corner supports the others, where nucleus colonies support brood honey production colonies when honey production colonies fail, or nucleus colonies can rebuild mating nuclei when they crash, or mating nuclei support give queens to the nucleus colonies that support the production colonies, or the production colonies give breeding stock to the mating nuclei, etc. Well, actually, I realized that in the center of that triangle should be brood factories, because brood factories support everything. Brood factories can boost honey production colonies, as Brother Columban did. Brood factories, harvesting brood from brood factories can make nucleus colonies without having to remove brood from your honey production colonies, thereby weakening them. Brood factories can boost cell building nuclei to incredible populations of nurse bees, making awesome queen cells. So the brood factories are really at the center of this whole system. My brood factories are in nucleus colonies, stacked nucleus colonies. They're in a vertical configuration as compared to a horizontal configuration if you were to use something like a Langstroth hive. In the vertical configuration, there's more queens laying in my double nuke box. So I have 2 nucleus colonies in the same footprint as a Langstroth. So I have 2 queens laying in that configuration. It takes less resources to have 2 queens laying. It's the same box, it's the same cover and bottom. It's just changed a little bit with entrances, so it's really the same amount of equipment. Ease of handling, I mean, it's so easy to pick these things up, but I think there's a real difference between vertical configuration and horizontal configuration when trying to raise brood to be used in the apiary. If you have say you have 10 frame Langstroths and you want the colony to expand onto new combs and raise more frames of brood. And so in order to do that, the bees have to expand the brood nest sideways to take care of those empty combs to the sides, warm them, polish them, get them ready for the queen. Well, how do they do that? They have to have a higher population. And so they're kind of stuck. You got to raise the larger population in order to populate those combs out there, but you can't populate those combs out there because you don't have enough brood to make enough population. But in the vertical configuration, in 5 or in my case, the cavities are only 4 combs, heat rises. There's only 4 combs. They don't have to expand horizontally. They can expand vertically. And boy, don't they expand vertically. And they just crank out the frames of brood. It's pretty incredible. So that's what I think. I think that this whole system based on vertical configuration, excuse me, I had a cough, is it's really helping my bees to crank out more combs of brood for me to be used. So what do we use these brood factories for? Well, there's boosting weak colonies, strengthening cell builders, starting nucleus colonies. So boosting weak and slow colonies. So suppose you have a weak colony, or a slow colony. I should call it a slow colony. I don't mean weak colony. It's just not expanding. The queen seems all right. and you want them to build up. And so what do? Rob frames of brood from honey production colonies, and thereby weakening them and thereby reducing the honey crop they make. Well, if you were to harvest a box of brood from the brood factories and put that box of brood on the bottom board below the slow colony, goodness, when those frames of brood emerge, the population explodes like you've dropped a bomb on it, and that's what it happens though. So that's how you would boost weak slow colonies, excuse me. So if you gave a box of brood, or you gave 5 or 6 frames of brood in the bottom box like this, think of the population increase in 2 weeks. Incredible. 2 to 3 weeks, you're going to double the population of that colony. Now they're going to be back on track. Now when you're boosting slow colonies, well, maybe there's a reason why there's a slow colony, and maybe it's the queen. So yes, I don't have time to mess with maybe it's the queen and maybe it's the colony and like that. I really only have one chance here. And so along with boosting the population, I'll give them a new queen. I raise queens, so I get loads and loads of queens. So, but where does that brood come from? That brood comes from brood factories. Nice emerging brood that there's just you're gonna explode the young bee population in that colony. Or how about strengthening cell builders? You know, this routinely, every cell builder we make this nowadays, we take a colony, a nice strong colony like the one on the left, and we add a box of brood on top. That green box is, it's got 2 frames of honey on the outside edges, but inside is 7 frames of emerging brood, emerging brood. And 10 days later, we graft into that. We set it up differently, of course, but we graft into what's going to be the cell builder. The green box is going to be the cell builder. And we put emerging brood in there. Now, when I first started this, I didn't use the emerging brood. I just used any old brood I could find out of my brood factories. Well, I was fortunate enough in, gosh, it must have been 2015, to be over in England on a speaking tour. And in near Lincolnshire, I met David Kemp. Now, David Kemp worked with Brother Adam for 10 years, from 64 to 74. And I was talking to him about putting brood in my cell builders. And he said, "You're using emerging brood, right?" And I said, "No, I'm just using whatever brood I get." He says, "No, you have to use emerging brood." And that really changed things. So what I'm doing in these cell builders is I'm growing nurse bees from brood harvested from brood factories. Now, there's other ways to add nurse bees to a cell builder, of course. I just like this one. I think Brother Adam had a really good plan, and I've been following it now for 20 years or so. And we built some amazing cell builders like that. Imagine putting a graft into that cell builder right there. Think about it. There's so many nurse bees in that colony, and they've got the field force, and they grow some really awesome queen cells. So what we're doing here is we're growing the population of nurse bees to increase the amount of jelly fed to the larvae, and that increases the quality of the queen cells. So it's all about the jelly when you're trying to raise quality. And we get queen cells like these this is probably day 5 or 6. They're just stuffed with jelly, great big pupae. Even on the day we put the cells in the mating nukes, they're still full of jelly. And how do you maximize the amount of royal jelly? Nurse bee population is increased by adding combs of emerging brood. And where does that brood come from? Of course it does. It comes from brood factories, not from donor colonies, not from production colonies, but from brood factories. Brood factories are dispensable, you know, they're disposable. You can use them up if you want. Or how about starting nuclei? Nucleus colonies. We make whole apiaries of nucleus colonies. These are all double nukes, 2 nukes in a stand, divided by, dividers, separate entrances. We make whole apiaries. In fact, this year, I think we made just over 300. We will usually make between 300 and 350. And all the brood comes from brood factories, every bit of it. We don't touch any, sometimes the overwintered nucleus colonies that weren't acceptable will build up, and because we're growing such, I'd have to say they're good stinky queens, because in the last 5 years we've made maybe how many hundreds of nukes, over 1,000 nucleus colonies, and I would say no more than 15 or 20 queens in that whole time were rejected by the nukes. When we take them out of the mating nukes, we put them in the new nucleus colonies. 20, 25 in all those nukes. So I know I'm on the right track when I say this. And where does that brood come from? Yep, you bet. It all comes from brood factories. So how I got started with brood factories, it's been a long, sort of a long deal, trying different things and making mistakes, and that's how it works, right? And so I needed brood for starting my cell builders and creating nucleus colonies. And so what could I do? Well, where am I going to get that brood? Well, originally, in my very early YouTubes, I talked about using the non-productive colonies in an apiary. Still a good way to go. So, you take a colony like this that's not making any honey, and you sacrifice it. You take all the brood and the bees out, you get rid of the old queen, and you make nucleus colonies. Well, that was nice, but you got to have non-productive colonies. So what if you don't have enough non-productive colonies? It was great when I wanted to make 100 nukes. Suppose I want to make 300 or 350. I don't have enough brood from non-productive colonies. So I went back to harvesting brood from honey production colonies. Well, here's a nice productive yard. Some of them have 150 pounds of honey on top, and so if I want to go in number 7 and pull a couple of frames of brood and make a nuke out of it, you mean to tell me I got to pick 160 pounds of honey off, take some brood out, put some combs back in, and put 160 pounds of honey back on top of that? That's not going to happen. That's what I did when I was 30 years old. I ain't doing that when I'm 70 years old. Forget about it, so that had to go. There had to be a better way than that. So then I started sacrificing the weak overwintered nuclei. So the strong ones would either get sold or put in production hive to replace dead outs, and there would always be the weaker ones. And so I'd move that division board feeder over to the side, give them 4 more combs, let them build up, and try to use their brood for making nucleus colonies. But I was having issues because there's a reason they were weak, and back in that time, back in whenever that was, late 90s or something, I had a real problem with chalk brood. And so a lot of these overwintered nuclei were weak because of chalk brood or because of a severe queen issue. And so I really couldn't get the nucleus colonies I needed from that method either. Well, so then I started saying, well, why don't I just take some of my strong overwintered nucleus colonies, let them build up, and harvest them for their brood? Okay, we'll try that. So we started with strong overwintered nuclei. Here's each of these stands has 2 nuclei. Look at the population already. You know, this is that dandelion bloom. You can see some dandelion bloom in the back corner. I haven't cut the grass yet, so it must be, you know, middle of May or something. The population is already exploding. Well, why don't I use those for harvesting brood? So that's what I did in 2011. From 50 overwintered nuclei, between May 9th and June 19th, I harvested 245 frames of brood to stock 35 cell builders. So each cell builder got 2 combs of honey and 7 frames of emerging. Probably wasn't emerging back then. It was probably whatever. So I hadn't met David yet, but and then I switched over to making nuclei. And we made 330 nucleus colonies. So altogether, if there's 2 frames of brood in a nuke, there's 300, there's 660, and there was 245. I'm harvesting something like 900 frames of brood out of these 50 overwintered nucleus colonies, which kind of blew me away. But that was it. That was the first time I said, wow, these things are amazing. And so the only problem was that those 50 nucleus colonies were sacrificed. I took them right down to nothing. I took every brood, everything I could get from them over a month period, and they were gone. What a waste. You know, so I had to come up with a better way than that. You know, I never got, after I changed to this new method in 2012, I never could redo those numbers again. I never got 10 nucleus colonies out of each one and some brood for cell builders, but I still had my brood factories. So I didn't sacrifice them. I just took what I could over time, and then I wintered them. And so every year now they're giving me brood, whole apiaries of nucleus colonies, all coming from brood factories. So I'm going to talk about the progression of brood and how and what we use in making nucleus colonies, in harvesting brood for whatever, for making nukes, for growing, setting up cell builders. And so I have a key here, a color key for the combs. So you can see there's honey, emerging brood, capped brood, mixed brood, brood in all stages, sealed eggs, larvae, whatever, nectar, pollen, empty combs, and foundation. And so these double nucleus colonies, when we make them up, this is how we make them. We give each a frame of honey. So this is a double nuke box, and the gray bar in the middle is the divider between the 2 nukes. It's in one 10-frame box with a divider. This one would have a divider over here. That one would have a divider on the backside. And they each get honey, a mixed brood, a capped brood, and an empty comb, and enough bees to pretty much cover the brood, maybe onto the honey out as well. That's really all they need. If you make them stronger than that, they get too strong too fast. And so a double overwintered nucleus colony would look something like this. This is after they come out of the winter in end of April. You can see what they have. You know, the one on the left has a couple of frames of capped and in the top, and a couple of frames of honey left. They got some nectar coming in, and they got some brood in the bottom box. You know, the one on the right is pretty much the same. They got a little less honey, they got a little more brood, they got an empty comb. So that's pretty much what they look like. They vary, you know, they vary in those, in what each one has. So we're going to add comb to the expanding nuclei. So we're building these things up to be brood factories, okay? But you can't keep them in 2 stories with 8 combs. Gosh, by the time midsummer comes around and we're taking brood out all the time and putting in combs or foundation, some of these they get up to 5, sometimes even 6 stories tall. You can't keep them in 3 and 2 or 3 even, unless you're there all the time, constantly, every week, pulling more out. Well, I've got 100 of them, so it takes us a little time to get to start at the beginning and 10 days, 12 days maybe, and go through everything and then start back at the beginning again. But you can see that we took this, and you can see what we did. We put a 3rd story on it. We put some empty combs at the divider, and we put some empty combs up in the center of the or the outside of the top box. Now we could have put all the empty combs in the top box, but remember, we're coming into a honey flow here, or nectar flow. And what happens when you put empty combs in the top box? Right, the bees put nectar in it. And well, I want brood. So in the center box, I put 2 empty combs, a capped brood and some honey. But those 2 empty combs are actually in the center of the brood nest. In a double nuke box, the center of the brood nest is not the center of the 2 combs in the middle box. It's actually the 2 combs against the divider, because the divider is the center of the brood nest of a double nuke box. And so you put these empty combs against the divider, and the queen jumps on them. Look, they're surrounded by brood. They got brood above them. They got brood to the side of them. They got brood below them. It's the center of their brood nest. And she jumps right on there and fills those up with brood. So I'm going to remove the left one. It's getting too confusing to keep track of and to explain to you what's all going on here. So let's just eliminate the left-hand side and start here. So this is an overwintered double-story nuke that we've added 4 empty combs to. Another story and 4 empty combs. And this is what they look like now. Now they're 3 stories high. Some are even, they grow so strong that they're now 4 stories high. Basically, they're 3 stories high. And now we're going into, we're probably into May sometime. And, early May is when we first start harvesting brood for cell builders. My traditional date is May 9th. That's the day we set up the first cell builders with that box of brood on top of the queen excluder. So we have to have these things really expanding by that time, so we can take a little bit of brood out, start slow at first, early May. And so this is what the last slide we had, harvesting, adding empty combs. So now they're going to build up. We're going to start taking, so there's, let's see. So if we're going to go in here and take some combs out for cell builders, we're going to start with some 3-story nukes now, and we're going to take them apart. The one on the left, with below the smoker, I'm going to take that one apart, and the bottom box is here. On top of the cover is the middle box and the top box. The top box is going to be quite a lot of nectar, honey. It's going to be rather heavy. The center box is going to have the bulk of the brood, probably, at least the 3 frames against the divider. The edge of the box becomes the divider against the other box or other super, right? And then maybe down below, there'd be 1 in here, and there'll be maybe 3 here, and maybe 1 up in the top somewhere. And so we're going to take brood probably out of the center box. You can see how crowded they get. So, Kate and Tucka are looking for they're pulling out frames of brood. I don't know what they're talking about. Probably, it's looking to see if it's emerging. making sure there's no queen, opening some cappings to make sure it's not purple eye, because, you know, purple eye isn't old enough. You need emerging when you're setting up cell builders. And so this is basically going to be a comb of emerging brood. There's some eggs in there, sure, but there's still emerging brood on there. We'll get a whole box of it, put it on top of the cell builder above a queen excluder, and in 10 days graft. So the slide on the photo on the left is the same photo where we added some combs. And then when we want to harvest brood, we have to look at what happened to the brood in the original box. So if you see in the top box, okay, remember it was put, empty combs were put in the top box at the beginning of a flow. So now those are going to have nectar or honey in them, okay? And then the mixed brood, you know, it was how many days ago? The mixed brood, or the capped brood next to it, that emerged while we were gone. And so then the queen relayed in it, and now it becomes mixed brood. The mixed brood got capped, and in that same time started to emerge. So now we have a frame of emerging there. The same one down below here. The two empty combs that we put against the divider are now full of mixed brood. The mixed brood is now emerging. So we're going to harvest those two frames of what am I doing here? We're going to harvest those two frames of emerging brood, and maybe one of the honeys. How can I get rid of this? Yeah, there. And maybe the honey. So there's two honeys in the top box. Let's take one honey and two frames of emerging. And then we'll put empty comb back where the emerging was, and move on to the next brood factory. And so now we've got a comb of honey and two emergings. We'll go to the next one. Maybe there's a frame of honey there, maybe not for our second frame of honey. And maybe there's another frame of emerging, maybe only one this time. And maybe the next brood factory will give us two, until we have 4 cell builders set up with 2 combs of honey and 7 frames of brood. And then we're done setting up cell builders for the day. Oops, let's see what's going on here. Okay, so then adding foundation is very similar to adding comb. So now we have these this double nuke box, these nukes that are now in 3 stories. We're starting to take emerging brood away already, but they're starting to get strong, and they're getting too strong, and we have to stay ahead of them. And one of the things I like to do with these is get some foundation drawn, as much foundation as I can get. So we're going to put a box on top or somewhere in there. We're going to put a 4th box on this, and we're going to do it with foundation. And so again, we can take some emerging brood out, and we put foundation in. And again, we put the foundation, some of it at least, against the divider. We want that foundation in the center of the brood nest. If we put it up on top, you know what's going to happen. They're going to draw it out and put nectar in it, but that's okay. That's all right. Nothing wrong with that. But we don't want it all up on top. And sure enough, you know, we'll pull out, there's 2 frames of emerging. We found 2 frames of emerging before we put the foundation on. We take them out, we put in empty combs in their place. And now we have this colony that's going to, it's now got 16 combs. It started the spring with 8. It's doubled its number of combs. You know, and each time we go through this 10 days, 12 days, we're finding additional combs of emerging brood. So you can see that that the empty comb, for instance, that was against the divider is now mixed. So she started to lay in it. And the mixed brood next to it, again, as before, The mixed brood is now emerging, and we can harvest that. So you can see that the mixed broods are now emerging broods, and we can harvest them. Empty combs are now mixed brood, and we can't harvest them yet. The foundation in the top box got drawn out, and they put nectar in it. So this is the cycle, and this continues on. And so they get up 4 or 5 stories high. And sometimes it's a struggle to keep ahead of them, you know, so that they don't swarm. So you have to be in them all the time. So once we have queens, after the first catch of queens, the first catch of queens is traditionally on June 13th. So on June 14th or June 15th, we start making nucleus colonies. We make as 50, 60 nucleus colonies in you know, right after that queen catch. And what isn't planned to be used gets sold, gets shipped out somewhere. And we're catching queens every 4 days. So from June 13th until about August sometime, we're catching queens every 4 days. And every 4 days, there's 60 queens or so scheduled for French hill apiaries to make nukes, and the rest get shipped all over the country. So making nucleus colonies. So it's the same basic thing. We don't need to have only emerging brood. We can have brood in all stages, you know, mixed brood. We can have cap brood. So the 4 boxes way over there on the right are starts, our beginnings, are their single nukes, just started, no queen yet. And you can see what we do. We take a honey, a mixed brood, a cap brood, and an empty comb. So what do we have here? You know, this one, this all the way over on the left, it built up into 5 stories high now. We're getting a bunch of honey on top. But still, it's the same progression. You know, cap brood becomes maybe emerging, or maybe we waited too long and it emerged and she laid in it, and the mixed brood becomes capped and then emerging. And so each of the new nucleus colonies will get a honey, a mixed brood, a cap brood, and an empty comb. Well, look, so we took a whole story away. We took it from a 5-story nuke down to a 4-story nuke. We took the top box of honey, 4 combs of honey, boom. There's 4 combs of honey for those 4 nucleus colonies, and so forth. And so each one gets the honey, the mix, the capped, and an empty comb out of storage. Well, sometimes the bottom box has empty combs, so we can use that. And if we don't have them, we put in empty combs that are in the shed. And again, we're making whole apiaries. This is a typical day, maybe 2, of making nucleus colonies. And we take them out and give them the queen. We give them the queen the same day. We make the nukes, done it mid-afternoon maybe. Take them to their yard, their dedicated nuke yard, give them all queens right then. So, you know, after all this, after all what we've been doing here, and, you know, putting the queens through her paces and just getting her to be a laying storm, an egg laying machine, some of them start to wear out. I don't like to have queens in my brood factories that are older than 2 years. I'll take a 2-year queen in a brood factory anytime if she's doing well. But once they get over 2 years, you know, you're risking having weaker colonies in the springtime, and I need really prolific queens. So we'll requeen them when they're 2 years old. And we pretty much always use push-in cages. Push-in cages are really great for requeening queen-right colonies. You know, you remove the old queen, and you locate the new queen under the cage with no bees, on nectar and emerging bees. And you put it back in the hive for 4 days. And you come back in 4 days and she's laying up a storm, and that emerging brood has just accepted her. And, you know, so the queens, the bees on the cage are, they're, you know, you can move them over with your finger. They're not being aggressive towards that queen. And when you pull that queen out, I'm telling you, within a minute, they formed a retinue. They've just closed in on her, formed that retinue, and she's been accepted. She's now a laying queen. She's not, you know, when the queen sits on my table in a queen cage for a day or two, she's no longer a laying queen. She's a mated queen. And if you want to remove a laying queen from a colony of bees, and you want them to accept a non-laying queen, you're going to have issues sometimes. But by having them under the cage, she comes in to lay, her abdomen swells back up again, she performs like a queen, she acts like a queen, and they accept her. So after making nucleus colonies, so we're done. If we start on June 14th, 15th, we're usually done making nucleus colonies by about July 10th. I don't like to go too far past that because our flow ends. And when the flow ends, there's no more drawing of foundation. They go into they stop their buildup. And so it's better. Our basswood flow is done about the middle of July. So if I can time my nucleus colonies to be made so that the population increases quickly enough so that you can take advantage of the basswood flow, that's what I'm shooting for. So I don't like to go too long. Yeah, you can go longer I could go longer into August and feed them, but I don't know. It's a losing proposition, I think. So after making nucleus colonies, you know, this is pretty much what they look like. This is before the goldenrod aster flow. We've given them foundation, you know, they've got empty comb. We try to take them down to take all their brood away and get them down to maybe 3 frames or 4 frames of brood. And so they would look something like this one over here. You know, the goldenrod flow hasn't started yet. So the goldenrod really is, if there's a decent goldenrod flow, they'll finish drawing that foundation out. Even though we've been pulling brood out of them constantly, the population is still increasing. And so we have to get them down before that goldenrod flow. We have to get them down to a manageable population and a manageable amount of brood. If you have too much brood in there, forget it. They're going to swarm on the goldenrod flow. So we have maybe 3 or 4 frames of brood. So there's let's see, and there's 1, 2, 3, 4 frames of brood. And that thing will build up into this colony on the right, excuse me, with all this honey on top, 2 boxes of honey on top. That's about 25 pounds in a box, and maybe some down below. And it's still got some brood there. So they're hatching out their winter bees. In a case like that, I would take that top box off. And they don't need all that honey. They only need the 1 box of honey, and the next one down will be after the goldenrod flow. That will probably be full of honey too. And that's plenty. So that's all they need. But that's what we're doing. We're building them up and we're tearing them down. And so now they're ready for harvest. So we get them down to 3 boxes. I like to winter them in 3 boxes high with a couple boxes of honey on top. Of course, there's always this. So one of the things that are really nice about nucleus colonies, especially these brood factories, is you're pulling brood out all the time. And where are the mites? The mites are in the brood. So you're pulling brood out all the time and giving it to cell builders or giving it to nucleus colonies. So that actually the brood factories usually have a pretty low incidence of population of varroa mites. But now you have to worry about your cell builders because they've been getting brood all summer long. And I'm not saying that the brood factories are heavily infested with mites, but they're there. So you have to deal with that. So last year in 2021 in the fall, we used oxalic dribble. We had a perfect weather for it. It was, gosh, November was just warm and the clusters were fairly loose and they were broodless. And I think it worked pretty well. I think it helped to some degree. I didn't do mite counts afterwards, you know, it's November. And then in the springtime, we used oxalic and glycerin pads. And, you know, I think they actually helped. I do. I found the colonies that were, that had an awful high mite load, it seemed like in the drone brood, you know, lots of young mites. And you would expect a colony like that, if it wasn't treated, to be crashing in July here. And, you know, we didn't treat in the spring with anything other than oxalic and glycerin pads. And I watched those colonies, and those colonies never crashed. In July, they had beautiful, healthy brood and beautiful white, pearly white larvae. And so, but we also had colonies that crashed. Same thing. So we just got to figure out why. What's the difference between those colonies? Anyway, so that's pretty much our varroa treatment at the present time. So now we have to decide, do they have enough food for winter? And so what we do basically is pick up the boxes. You know, I can't weigh them because there's 2 nukes in a box. And so we just pick up the top box or 2 and say, is it light? Is it heavy? Is it medium? Is it, what do you think? What is it going to take to take that 3-story nucleus colony with 4 combs in a box through the winter? And they'll need the top box full of honey, the middle box almost full of honey, and the bottom box maybe with a little bit. So we pick up the top box and it's heavy. So you don't need any syrup for that box. You pick up the next box. Well, it's about half honey. Okay, so we give them a gallon of syrup. If you pick up the top box and it's really light, and the next one down is definitely going to be light, then they might need 2 or 3 gallons of syrup. So that's pretty much, it's an estimate. This is sort of the progression of a brood factory through the season. This is in 2021. It started out in March, and we just look at it to see, is it alive? Does it have worker brood? Does it have enough food to eat? Close it back up. Okay. One on each side, right? Okay. Then we looked at about, I don't know what that says, 422 maybe. The one on the right, on the left had eight frames of bees. The one on the right had 4 frames of bees. Difference already. On the 13th of May, the one on the left had 8 frames of brood. So that 8 frames of bees turned into 8 frames of brood. Had a green dot queen. We took a frame of brood away. Gave it an empty comb somewhere in the middle of their brood nest. The one on the right had 5 frames of brood. Somebody didn't like the looks of her pattern or whatever. It said requeen. 5 days later, we gave this the one on the left the 5th box. So they're exploding with bees. Look, of course they did. They had 8 frames of brood, and now they had 7. But you know, so they just fill those things up immediately. And no, we didn't take anything from the right. It says requeen again. So there's something going on there. You know, and you can see here on the 14th, we took 2 frames of brood. But then on the 14th, the one on the right said, oh, wait a minute, they need to have some brood taken away. So we took 4 frames of brood away because we were way behind already. And so on and so forth. You know, here's one minus 3 frames of brood and a frame of honey. Obviously, we're making nukes on that one. And so forth and so forth. And pretty soon, when we get later in the season and we're just about done making nucleus colonies at the last round or something, we take a lot away. We took a nuke and a half away On the 20th, we're looking for requeening. You can see that the queen on the left nuke was okay. She was doing well. She performed well through the season. Her pattern was good, no disease. We'll let that one go for another year because it was only a 1-year-old queen, as it says down here, green dot queen. The one on the right, you know, since it was twice said requeen, we requeened it. So we killed the old queen. And we put a daughter of breeder 84 under the cage and then release them. And after we get done, you know, requeening, then we start thinking about how much food we need to feed. We start picking up the boxes and hefting them and seeing what they're like. And, you know, once the goldenrod flow is done, that's pretty much it for us. So as soon as you can't smell the goldenrod flow anymore, we estimate how much syrup. So in 2021, especially up in the northern part of my range, we had a terrible drought. I'm sorry, Randy, but not like yours, I know. But we did. For us, it was really dry and there was no nectar coming in. And so they didn't make anything. So a lot of the ones up in the north part, they needed 3 gallons or even 3 gallons and plus. So we needed to feed a lot of syrup last year. So this is how we do it. You know, each nucleus colony has a little inner cover, a half width inner cover, and a feed hole. I put a couple of shims down, 3/8 shims, and put syrup cans on top with holes in the cover. You can put 2 on at a time. They meet over the hole. The bees will go up there and suck all that syrup right out of there. And in a few days, they'll take down 2 gallons of syrup in surely less than a week. Then we put an empty box around it to hide it from the weather. And sometimes if there's a crack, you put some tape on it to keep the robber bees away. And then we wrap get them ready for winter. So the feeding is done. They've been treated for varroa mites. Now we have to wrap. And we have an upper entrance. For instance, the second box from the left, you see that upper entrance hole. That's the upper entrance, a 3/4 inch hole. Now, a lot of people say, oh, you shouldn't have upper entrances or you know, upper entrances cause more honey to be eaten or whatever. Well, this is typical. This is our first snowstorm. And the bottom entrances at the bottom of those nuke boxes, they're closed up with snow. So what are the bees supposed to do if they have a possibility of a cleansing flight in our winter? I mean, our bees go 4 months with no cleansing flight. And when it presents itself, they need a cleansing flight. And they got to have it. And that upper entrance gives them the chance to have that cleansing flight. But also, it releases some of the moisture. There's a huge amount of moisture in this given off by these wintering colonies, and they have to get rid of it somewhere. And if they don't, it just builds up inside. You have puddles in the bottom, the water running down the side walls. So for 2 reasons, we have an upper entrance. So this is this is a brood factory yard in the cell building yard itself, all wrapped up for winter. So this is what we do. We put tar paper around it, black tar paper. It's not for insulation. It's for solar gain. So on a nice cold winter day, wow, you ought to feel that the heat that's generated by that black paper. And I know that it somehow moderates that cold temperature inside the hive. Bees can maybe go out and pick up some honey and bring it into the core of the brood nest again. You can see I'm stapling it. I'm just stapling it on. It stays. It's got the upper entrance. I always tie the covers down because covers have a way of blowing off here in the wintertime. So, you know, in a couple of minutes, in just very few minutes, look, the bees are already looking out the hole. I mean, so they're very aware of that entrance and they love that entrance. In fact, they'll prefer that entrance to any other entrance in the hive. So we have to close them in the spring and try to get them to start using the bottom entrance. So what exactly is it that we're doing with these bees? You know, if you think about it, we start them in the spring and we build them up, and we add some more comb, and we build them up some more, and we harvest brood for cell building and replace it with comb or foundation. And we get them built up from maybe 3 boxes tall to 5, 6 boxes tall. These enormous colonies that have huge populations. And then we start making nucleus colonies. And then to make nucleus colonies, you know, we're taking away combs of brood and bees and honey. And at some point, we don't replace those combs. We just take and we knock them down and we knock them down. We try to get them down into 3 stories again for winter. Sometimes we got to go to 4, but try to get them down. So we're building them up and we're knocking them down, and we're building them up and we're knocking them down. And then we winter them. And they become a perennial brood factory. We don't sacrifice them. I don't sacrifice them anymore like I did in 2011. I take what they give me, and then I winter them the way they are. And in the springtime, they're ready to go again. Build them up and tear them down. And all this from a single-use resource hive. This is what I'm trying to get at. You see how valuable these things are. You see what they support, what they supply to my apiary. And so many people have said, oh my God, this works so well. Yes, it does. So you give it a little try, you know, you don't have to have 100 of them. Have a few, try it out. It takes a little time to learn, but boy, the resources that they will give you to support your apiary is just incredible. And it all comes from brood factories. Anyway, thanks for listening.
Well, thank you for presenting, Mike. This was really incredible. It's like you're a choreographer out there with your combs. I mean, it's just so much going on and to keep track of. And I think we have a couple of questions that look more like clarifications on your equipment and design, if
Sure.
you don't mind.
Not at all.
So one individual has asked here, what kind of center divide do you use in your bottom boxes, and how do you build it into the box?
The center divider is just a 3/4 inch pine board. If you don't have a deep enough one, you can shiplap it so that it's, you know, there's no crack. And it's rabbited into the front and back wall, halfway through the box, so that the divider goes all the way through the frame rest. So there's no way for the bees to get around the frame rest. See if I can get this thing here. No. Ah, there it is. Yeah, so there's no way for the bees to get around that divider. And the divider on the bottom board is a cleat, a 3/8 cleat, and the divider goes from the bottom of the box to the top of the box. So when you put the box on the bottom board, the divider touches the cleat. And then on the top of the box, you either have a bag or you have two little a half width inner covers that meet on the divider, so the bees can't cross over. And then as you put supers on, the inside wall of each super becomes the divider. So the nucleus colonies are separate all the way up to the top.
I think that, thank you. And I think that also answers one of the next questions here is, are the stacked nukes divided by queen excluders or all individual? And I think, I don't know for sure whether or not they are wondering well, I guess the obvious answer is that they're not
They're
separated
no,
by
no, they're
distinct.
absolutely individual. They're separated. Some people have tried using queen excluders on top and then honey supers above. And yeah, you can harvest honey, but there's issues. First off, if one side wants to change the queen, forget about it. They're not going to change the queen because they have a queen on the other side of the queen excluder, even though it's up at top. I have harvested frames of honey. The bees will go up and work side by side, doesn't matter which nuke they came from. And then they'll store honey in supers. And then when you take the super off, you put a bee escape under it or something, and they'll go down into whichever nuke they go down into. It doesn't matter. They never fight, but there's issues. They swarm, they just swarm too much. They get to be such enormous colonies that I think it's better to make honey and honey production colonies than use these nucleus colonies as nukes or as brood factories.
Okay. I'm glad you brought that up. We have a rather good beekeeper up here in Alberta in Canada who has modified your system. He's really thrilled about what you do. He's not a queen breeder. So he's been running the nukes all the way up, and then an excluder, and then the honey supers on top. And it could be because we have such a different sort of season and climate.
That's for sure.
Yeah, and he's found that very successful.
You have
And
one flow?
we have one very long flow, really.
Yeah,
it's
from canola?
a lot of sweet clover and alfalfa too.
Oh yeah, that too. Okay.
Yeah, so it's a long continuous flow. If the weather's good, the bees are making honey all the way from about the 20th of June until about the 20th of August. All we need is sunny days, and we usually have enough moisture in the area so that it doesn't. So he has a long honey flow, and he has typically averaged 250 per hive, but that's per double colony, with two queens. And anyhow, I thought I would tell you that, so because it is a different area. But
do you find that if you have just standard honey production colonies, that they produce significantly less than that 250?
The
I
Alberta
was up
average.
there. I've been in Winnipeg and Kamloops, and people, the big beekeepers are talking about 250 to 300 pounds on canola and things. And whoa, I mean, I couldn't imagine having 250 pounds worth of supers on top of my double nukes, because I need a ladder to get to them.
Yeah, he does have them stacked up. Sometimes parks his flatbed nearby.
Oh yeah, for sure.
The average for Alberta is 140 pounds, and
Oh, okay.
about 70,000 colonies go into canola, so that does reduce the overall average. We have about 350,000 colonies here in the province.
Wow.
And the typical beekeeper, the typical honest beekeeper is making 180 to 200 pounds most years. So his 250-pound average over the years is slightly better. But what he really likes about it is that he feels that he never has a queenless colony, because they're talking to each other. He doesn't have knockouts. And that seemed to be one of his advantages. Now, it's not my operation, so and I don't know exactly any of the idiosyncrasies, problems, or issues that he's encountered over the years, but he stayed with it, and he credits you as his source.
Well, thank him for me.
Yeah, no, he's very happy with the system. So along that same line, I think I saw a question here. Yes, here it is. How do you deal with uneven buildups? And this would also apply to my friend, who, one side is doing really well and the other side is doing weak. Well, you showed quite a bit of manipulation to help solve that problem. But
right. So I may, if we have say we have one that's in 4 boxes, and they're doing really well, and the other one isn't doing so great, so they're only in 3 boxes. And so we either put the inner cover on top of the 3rd box and put an empty super on top, so that the top of the hive stays level, and you put the lid on. Or sometimes if there's a bag, we use grain bags as inner covers. Sometimes we'll fold the grain bag in half and put it on top of the weaker one and put some empty boxes on top of it. So
Mhm.
that's what we do. And then if it needs help, we'll give it help. A lot of times it's just lagging behind and it will get built up to full strength.
Right, right. And you gave a good
But some of
example
them are
of that.
just so outproduce the other ones that there's no hope that the other one will
Mhm.
ever catch up to the very stronger one. So they stay at 3 stories and the other one goes to 4 or 5,
Okay. Yeah. Yeah. I think that answers that very well. Um, Mike, do you prefer the entrances of your nukes on the front or the side of the hive? And I noticed you have a mixture.
I do. And when I first started, I was wintering them in one box, 1 story high, two nukes in a box with a movable divider. And then I'd winter them on the inner cover of production hives. So there's no contact between them, but just seemingly the nukes would get a little heat from the production hives and we'd wrap them all together. And what was I going to say? What was your question again? Sorry.
No, it was the difference between front and side entrances.
So
on your,
yeah, so I didn't feel I could have the entrances in the front because the top entrance of the big colony of the strong colony would be right there next to so I put them on the sides, but I hate them on the sides. I hate them on the sides. So now everything I've made in recent years has been front and back.
Mhm.
And if I've used some of the bottom boards they have 4 entrances. We put duct tape on one and eventually those bottom boards fail and we get rid of them. And so we're changing them all over to front and back. It fits
And
better in the apiary to have them front and back.
yeah, and your drilled winter entrances were
It's
all on the
always on the
front.
same side as the
Mhm.
bottom entrance.
Mhm.
So if you close
Okay.
the top entrance with tape or something, they just crawl right down and go in the bottom entrance. And they don't,
Mhm.
like when it's on the side, if you do that, then they got to find their way back and, I
Yeah.
don't know, better.
Yeah, yeah, yeah. Question here is, did you say that your visits and brood removals are on an 8-day cycle?
I would say more than that.
More
More than
days.
that, probably 12 or something like that.
Mhm.
Probably 12. We work on a 4-day cycle. So we're setting up cell builders every 4 days. We're grafting every 4 days. We're catching queens every 4 days. So and so three times 4 days is 12. So we try to get back to the original starting point of the very first brood factory we took from 12 days, 2 weeks later, and then we pull more brood out and we make the progress around the 100 or so, and they're in different yards. And then we'll start over again at the beginning. So it's going to probably be 12, more like than 8. 8's too soon. Yeah, 8's too soon.
So 12 is the more typical. And you mentioned the basswood flow. I don't know if you have tulip poplar up your way either,
None.
but
No.
no. So during the basswood, then, do you sometimes remove honey for extracting?
We usually wait till the end
Mhm.
when it's all done.
Okay.
Rather,
Okay.
and then we harvest it as a blend. We don't really try to harvest varietals. There's just too much going on to
Okay.
take honey off and
Yeah.
extract when we're trying to raise queens and make nukes and all this other stuff and requeening. Oh my gosh, you know what it's
Okay.
like.
Yeah. Yeah, I do. Some experts, that would be you, recommend comb rotation for the health of colonies to reduce contamination buildup. Michael, do you notice any health differences between brood factories with constant comb change and honey production colonies that don't get as much comb rotation?
I mean, old combs definitely have a reservoir of pathogens, obviously, and pesticides, obviously.
Mhm.
It's hard to pull out a nice worker comb that's just a perfect worker comb. And the queen just loves those combs so much. And it's really hard to get rid of those. But there does come a time when there's too much drone, or the comb is misshaped, or it's got entombed pollen in it. You know, if you know what entombed pollen is, a lot of people don't know what entombed pollen is. It's pollen that the bees have decided there's something in that pollen that they want to keep away from their sisters, and they cover it with propolis. It's not shiny old pollen. Old bee red and shiny is shiny old pollen. It's not that. It's covered with propolis, and you can dig it out. Many more often than not, it's brick red. Don't ask me why, but Jadzak from Maine sent some to Penn State, and it was just loaded with a list of pollutants. And it's, you know, it's not just our country. I was in Veracruz, Mexico, looking at hives of bees in a coffee plantation, and darn, wasn't there just tons of entombed pollen. And I opened it up, and what color do you think it was? Brick red. And I don't know why. I'd love to know why, but
That's
you know, so
fascinating.
if there's entombed pollen, that comb gets taken out and rendered or burnt, you know, whatever.
Yeah.
But other than that, you know, unless it's an ugly comb, I mean, sure, we get rid of those.
Yeah. Yeah. And of course, when the molting starts to shrink the size, I suppose, too.
Right. Yeah.
About how many pounds of honey is required to overwinter a brood factory?
I
And
would
what
say
should
that,
they weigh?
I would
what should they-
say, well, it's hard for me to weigh because they're two in a box, and I can't weigh one without, you know, the other. So somebody that works for me says that he likes the 3-story nucleus colonies to weigh, you know, the separate ones, they're separate, 85 pounds.
Mhm,
So if that helps.
mhm.
And the 2-story, I think he said 65 or 70 pounds. But for me, you know, I don't weigh them. So I want the top box to be full of honey.
Mhm.
And that's going to be around 20. If there's 4 combs and it's 6 pounds in a comb, there's 24 pounds.
Yeah.
And if the next box, the middle box, should be full of honey also.
Okay.
That's plenty. I mean, or even if the middle box has 3 frames of honey, and maybe the one against the wall has some empty cells, and the bottom box has maybe the outside comb has some nectar or honey in it, and then there's some pollen, and that's about what I look at. You know, I pull the top box off. Wow, that's heavy. And I look down below, and it's burr comb full of honey, and you stick your hive tool down in there, and all you come up with is honey. That's enough. That's enough.
Well, we're keeping you up here almost till midnight. So we have 2 more questions in there, and they're kind of related. So if you don't mind those last 2.
Not at all.
Thank you. So it's related to genetics, your stock. So
Mhm.
so they want to know what genetics do you have now, and are you using much VSH? I'm assuming they just want to know like what stock you've got.
Right. I guess I have my own. I've been doing this for a while. You know, of course, like everyone else, I bought Italians to begin with. And then the old acarine came through, tracheal mites came through, and pretty much wiped out all the yellow bees. And at the beginning of that, it was recommended by an old guy that I should be buying Buckfast. And so I bought Weaver Buckfast from R. Weaver down in Texas. And I'll tell you what, that pretty much saved me from the huge tracheal mite losses, because Buckfasts were so resistant to tracheal mites. But then they seemed to have chalk brood. So I requeened all the chalky hives with a really hygienic strain that I bought. And wow, in a month, I couldn't find any more chalk brood in a month. And that was the beginning of my breeding program. That was in about 98, 99. And so then I just selected from the best of those different lines, and You know, I've brought in very little stock over the years. I'd trade a breeder with somebody, see what their stock looks like, and I'm often disappointed either by their temper or their swarminess or something, chalk brood or something. So I haven't got anything in years. So we've just been I've been selecting from, you know, colonies that have performed well, colonies that have the temper, the coloration that I like. It's hard to keep away from Italian stock here because so many people bring in uncounted numbers of bees from down south, you know, nucleus colonies all around me, packages all around me. So we are trying to select by color a little bit. We've got a breeding program now this is the second summer. We're working with the University of Vermont Bee Lab. And 2 summers ago, we identified 64 possible breeders. Did samples, hygienic, you know, frozen assay, brood assay, you know, nosema, and all the records I have on those colonies over the years. And, you know, if there was any issues anywhere, if the population, the Burrow population increased, they were off the list. And we got it down to 5 really nice ones. And I picked 5 of my own way without selecting for everything, so we could have controls. And then this summer, we had 10. We had 5 of my own and 5 from that the program picked. And then then we wintered them over. I gotta get my plug in, and wintered them over. And then this year we repeated the tests again. We made from those 10 breeder queens, we made 100 nucleus colonies in 2 different yards. And we had of those 10, we had 5 of each in each yard. There's so 50 nukes in each yard. But also, and we did the same thing, the frozen brood assay and all. But this year we got something really interesting. It's called the unhealthy brood odor assay. Have you heard that? Have you heard about that?
No.
So a woman from North Carolina, maybe in Greensboro, North Carolina State, Greensboro, identified some volatile chemicals that are given off by unhealthy brood. Not dead brood like frozen brood assay, because that's just picking up death. This is unhealthy broods. This means high mite load or high virus load, even if there's no mites. And so she isolated it, had somebody you know, make it. And so you put a little ring, plastic ring on the comb. It has to be purple eye, can't be emerging, it has to be, you know, white or purple eye. And you spray it on there, and it's about 20 cells. It's only 20 cells. And you put that comb back in the hive for 2 hours, not 24 hours like frozen brood assay, 2 hours. And then you look to see what's been uncapped. And it's pretty amazing. Oh, wow. Some of them have uncapped it in 2 hours, have uncapped it all. And so we're hoping that this is going to be an easier way to detect high VSH colonies, rather than the typical, you know, look at 1,500 cells and, oh my goodness, who's got time to do that? You know, we got to take care of the bees. So we're hoping.
That work?
We're hoping it does. I mean, it's really you know, you can see the difference between some breeders. Some daughters of some breeders are nothing. And some of them, I mean, it's just open. It's all uncapped in 2 hours. It's incredible.
Wow. Well, thanks for letting us into your operation. I don't know if anyone on the panel or anyone else wants to add anything, but we really appreciate hearing from you. And it was a great story and quite an operation that you've built up over the years, and so much that you've learned and so much you've shared with everybody.
Yeah, you know, it's really my focus now is, you know, sharing, is education. You know, that's really key for what I do from now on, other than this breeding program. The breeding program is keeping me in bees. I'm already old enough. I don't know, but so I can't do what I used to, but I've been around the world, and it's just really nice to be able to talk to people and then have them write me back someday and say, wow, you know what? I tried that out. Wow, that really does work. So, that's been just awesome for me.
Well, it's been really appreciated by all of us. So now that it's almost midnight where you are, thank you for this.
Oh yeah. It's only
Oh, yeah.
11:30.
It's been great.
Michael and Dave, thank you so much. That's great.
Surely. Yeah, we'll see you all again.
Thank you.
Have a good night.
Good night. Yes. Thanks.